What comes to mind when you hear the word diabetes? Perhaps insulin popped into your head. Insulin is one of the most common associations with diabetes mellitus, a disease of high blood glucose, and it has long been the focus of diabetes research. However, insulin does not act alone in regulating blood sugar. It has a critical partner, glucagon. While insulin release from pancreatic beta cells lowers blood sugar, glucagon from pancreatic alpha cells raises blood sugar. We know that insufficient insulin production leads to high blood glucose and diabetes. But what role does its partner glucagon play? Are alpha cells, like beta cells, dysfunctional in diabetes? A major problem in answering these questions has been the lack of accurate models reflecting human pancreatic islets, which house alpha and beta cells. A critical advance has been the technique of transplanting human islets into mice. This has allowed us to study human beta cell function and insulin production in vivo. However, we run into a problem when trying to assess human alpha cell function because unlike insulin, mouse and human glucagon have identical structures and are thus indistinguishable. To overcome this limitation, my project uses a groundbreaking model in which the mouse glucagon gene is knocked out. This allows us to study solely human glucagon after transplantation of islets into mice. So with this model, we've asked the question, how does hyperlipidemia, a key risk factor for type 2 diabetes, impact beta and alpha cell function? In other words, what is happening on both sides of the seesaw? We found that mice fed a high-fat diet had reduced stimulated insulin secretion. But more notably, we saw they had significantly increased stimulated glucagon secretion. Our findings indicate that excess fat not only impairs insulin secretion, but also enhances glucagon secretion. We next looked at potential mechanisms driving alpha and beta cell dysfunction. We found reduced levels of two key beta cell proteins, but did not see any changes in the unique alpha cell protein ARX. We're now assessing other genes that may contribute to alpha cell dysfunction. So what are the clinical implications of my research? Well, we are already seeing the benefit of adding glucagon-targeting drugs to diabetes regimens. My research is critical for understanding why these drugs are clinically effective, and moreover, for developing novel targeted therapies that will advance diabetes care. Thank you.